All right, so what we're talking about here in chapter 19 is the control of gene expression. And this is part one of probably a three-part series. And so what I've put here on our front slide is what they call a gene array. And so we're going to look at one example of how this type of data is generated later on in this presentation. But what you see here, where it's green, these particular genes have been turned on, and where it's red, those genes are turned off. So this is mega data. So you might have heard of bioinformatics, which is a field of science where they're taking all of this mega data here and trying to make sense of it. So they're trying to figure out, what are all of these genes and what are they doing? So we're going to kind of take a look at this, but I just thought this is a nice visual to show us, the amount of data that we're looking at inside of our cells. All right, so again, this is chapter 19. And what we're going to focus on today is looking at the chromatin, so this relaxed state of DNA during interphase, and how we're able to essentially remodel it, or open it up is another term that they use, so that we can actually access the genes inside of this really big molecule. And then I'm actually going to back up and we'll do section 19.1. My goal is here to do 19.3 and 19.4 on Monday. And then Wednesday, we will do 19.6, which is a regulation, defects and cancer. And this will then bring us nicely into looking at control of our cell cycle and how mutations there can also lead to cancer. So we're going to end the semester off kind of looking at how different types of mutations and defects give rise to different, cancers. We are not going to cover section 19.5. It's a really tiny section. If you want to look at it, you can in your textbook, but I'm not going to hold you guys responsible for that content. All right, so I thought we would start off with just really trying to wrap our minds around how big is this human genome. So you guys are all pretty comfortable at this point with what DNA is. You know it's made up of a sequence of nucleotides of A, T, C's, and G's, and that it's read in, a three-letter code that gives rise to proteins, right? We've been looking at that over the last several weeks. But we haven't really talked about how large this DNA molecule is in the human cell, all right? So DNA in humans is approximately 6 billion base pairs long. And so if you were to stretch that out, it's roughly 2 meters, I think it's like technically 1.77, or 6.5 feet long, all right? So that's pretty long, right? And if you think about that as like a string, right, you've got to fit that inside the nucleus of every single one of your cells, all right? The diameter of the DNA is about 2 nanometers. And so I put here roughly what a nanometer is with respect to a meter, all right? So if a meter is roughly like 3.25 feet long, we can all kind of maybe visualize that. This is really small, right? So you've got to move your decimal point over 9 places to figure out, the diameter of this DNA. So it's extremely long, but its diameter is really tiny, okay? But if we look at the diameter of the average nucleus inside of a mammalian cell, it's about 6 micrometers. So a micrometer is a little bit bigger, as you can see, than a nanometer, but still very small. And so what we're trying to do, and I calculated here for you what the volume, if we assumed that the nucleus was a sphere, the volume of that nucleus is about 37.3 micrometers cubed. All right, so it's really tiny. And if we've got this string that is 6.5 feet long, how on earth are we fitting that inside of this teeny tiny little nucleus? All right, and I was gonna try and find, I just ran out of time though, to find a string that was like 6.5 feet long, right? And trying to fit it inside the smallest little thing I could find here at the house, to see if we could get it to fit in there. Because I think you would see it's, it almost seems impossible. All right, so our question, our first big question of today is, how do we fit all of this DNA inside of the nucleus? And just another fun fact before we look at that, if you were to take all of the DNA from every single one of your cells, it would stretch all the way to the moon and back, I think like 600 times. So that's a lot of DNA. All right, so as we start to look at the control of gene expression, some of the things that we're going to have to think about, is how do we know which gene, is going to be expressed, right? Which gene are we going to transcribe? So the regulation of this process is a lot more complex in eukaryotes than in prokaryotic cells, due to the complexity of, having these larger cells that might be, either a single-celled organism, or in most cases, eukaryotic cells have formed together to form multi-cell organisms. And so how does this process work? So one of our new vocabulary terms is called differential gene expression. And what this basically means is that different cells are going to turn on different genes at different times, right? For the most part, and I know there's some recent research that has come out that kind of, it's a little bit different, but for the most part, we can think of all of the cells in our body containing the same DNA, right? So why don't all of these cells look the same? Why don't they all do the same thing? All right, and part of that answer refers to this differential gene expression. And so the ability of our cells to be able to turn different genes on and off is what helps create all the different cell types in our bodies. It helps them to arrange into specific structures that form our tissues and organs and organ systems. And then this differential gene expression also helps them to coordinate the activity that has to occur between all of these different cell types, right? So this is a very, very complex process. So I want to take some time to kind of dig through this and see how all of this works. All right, so let's start off with looking at our DNA and seeing how, first of all, we're able to fit it inside of the nucleus. And then based on that, we'll then have to figure out how such a compacted structure is going to be able to allow these different genes to be transcribed. Okay, so what we're looking at here in the picture on the right is an illustration of chromatin. So if you can recall the difference between chromatin and a chromosome, chromatin is when we have that relaxed state of DNA, right? So during interphase, when a cell is not dividing, the DNA is what we call in its relaxed state in this form of chromatin. But unlike that illustration I showed you guys, I don't know, probably a couple months ago now, it was before spring break, we had I had a picture of a bowl, right? And I threw a bunch of DNA in it and said, hey, this is kind of like, a bowl of spaghetti. It's not quite that disorganized, because if chromatin was really that unorganized, we wouldn't be able to fit it inside of our nucleus. So what we see here in this picture is that DNA, represented by this red color here, is going to be wrapped around some very special proteins known as histones, represented by the green ball structures. All right. What this interaction allows is a much closer, tightly packed DNA, right? So if you recall, DNA is a negatively charged molecule, right? And if you have a bunch of negative charges next to each other, right? Are they going to want to be close to each other, or are they going to repel each other? So if you think about a magnet and you're trying to put, two negative ends of that magnet together, are they going to attract or are they going to repel from each other? Does anybody want to give a guess in our chat box what they're going to do? So if you're trying to put a bunch of stuff that's the same charge, and again, think about magnets. Are they going to repel or are they going to attract? Yeah, thanks, Tyler. They're going to repel from each other. So this very large 6.5-foot long DNA molecule, if we're trying to wrap it and squish it close together, energetically, it's not going to want to do that, right? It's negatively charged. And so it's going to repel slightly from itself. So these histone proteins actually contain a lot of, I believe, cysteine and serine amino acids, which give it an overall positive charge. And so this allows the DNA, as you can see in the bottom picture, to wrap around these histones very tightly. And so this allows us to pack it really close together. And as I have mentioned here on the slide, there's about 200 bases of this negatively charged DNA that's going to wrap around a core of 8 positively charged histones. All right, and so that's what we're showing in this bottom picture. And we're going to refer to this particular structure as a nucleosome. All right, so there's another, I've tried to highlight the vocabulary here in yellow. So histones are the individual green balls, and then this complex that we've built here with the 8 histones and the DNA, this is known as a nucleosome. All right, so that's the first step in kind of, getting this to pack closer together, but it's not enough. All right, so what we see here in this picture is that there is going to be a stretch of DNA that's not wrapped around these histones, and this is known as linker DNA. All right, so I didn't write that over here in the text portion, but the linker DNA is the DNA that kind of links these nucleosomes together in that entire, strand of chromatin. We're going to see that there's an additional histone protein or specific histone protein called H1. And this is represented by these blue structures located here. And the function of this H1 protein is it's basically attaching like the linker DNA to the nucleosomes. And then it allows for additional folding of these histones into what's known as the 30 nanometer fiber. And it's named that because the thickness of this is now 30 nanometers. And so this fiber then can fold, as you can see here, in and upon itself. And eventually, when we get to cell division, so in that synthesis phase, and we create individual chromosomes, it will compact even more tightly. All right, but as we look at this right here, this would still be considered chromatin, and the actual chromosome would form only during cell division. All right, so this packing is what allows this 6.5-foot length strand of DNA to fit inside of our, 6, what was it, micrometer nucleus. All right. So the 30 nanometer fiber then attaches to some other proteins that are known as scaffold proteins, and that's really what holds the whole chromosome in place. When the chromosomes condense before cell division, we'll see that they become even more tightly packed. So this up here, I should maybe refer to this as chromatin, because again, it's going to stay as a single strand of DNA really until we get to cell division, where then they're going to break into pieces and form chromosomes. But this will make a little bit more sense, I think, when we look at the next slide, because they've actually been able to image where each of the 23 chromosomes are, even when we're in this really long strand of chromatin. So it's a really cool picture. I'll show you guys that in just a second. So chromatin's elaborate structure, so using these histones, forming those nucleosomes, and then being able to form that 30 nanometer fiber is really what allows it to fit in the nucleus of a cell. We're also going to see that the way it folds, so if we go back, oops, go back to this slide, how it's folding here is actually going to play a key role in also regulating which genes are going to be turned on and turned off. Okay, so this next slide is a really cool picture. So this picture here on the right kind of looks like a giant jelly bean, is what our chromatin, so this is human chromatin, what it kind of looks like inside of our nucleus, right? So it's all kind of jumbled together, and they've been able to map out exactly where in this jelly bean looking thing each of the 23 chromosomes is located. So right here, like we can tell that this particular DNA came from a male because we've got our X and Y chromosome here. And so what's colored here in this like teal color would contain all of the genes located on the Y chromosome. And we can see here in yellow, it's a much bigger area. This is where all the chromosome or all the genes are located for the X chromosome. So really, really interesting how the chromatin folds, and that scientists have been able to even identify, where these chromosomes are now in that big massive strand when it's all kind of jumbled up like this. So the important take-home thing here is that each chromosome is really occupying its own distinct region inside the nucleus. And so when I look at this, I also kind of think of our brain, right? So our brain is kind of like this jumbled mess of neurons, but each section of our brain also is, has a very specific role. And so each of these regions is going to have a very specific role in gene expression. Portions of these chromosomes then are able to, what our textbook described, is kind of like fold out or pop out of this structure here so that it can become accessible to our RNA polymerase so that we can actually, generate the messenger RNA transcript. Because when all of our chromatin is this tightly packed, we're not able to actually transcribe anything. And so the location and layout of where each of these genes is located is actually important. So those that are located more on the surface, we'll see are ones that are probably going to be more actively transcribed throughout the life of that particular cell. All right, so now that we know how this extremely large DNA molecule can fit inside the nucleus, our next big question is, how is the cell then able to go in there and identify which gene it needs to transcribe, right? There's got to be a very systematic way to do this. So the answer is going to lie in the chromatin structure, and what we're going to do is we're actually going to alter that structure to activate certain genes. So in our previous chapter, when we were talking about transcription, we mentioned something about a promoter, right? So DNA has promoters, right, which are regions along the nucleotide sequence where the RNA polymerase, recognizes it and binds to the DNA to start transcription. So how are we able to access those promoters when all of this chromatin is so tightly packed? What we need to be able to do is we have to decondense that particular region of the chromatin so we can expose that promoter sequence on the DNA. And Weintraub and Grudine were two scientists that tested this process to see if this is exactly how it worked. And they used an enzyme that's able to cut DNA at random locations. But the key here is that that enzyme cannot cut DNA when it is tightly condensed. And so they were, looking to see if it can preferentially cut genes that are actively being transcribed. And so just a quick schematic of what this kind of looks like, or what that context meant, is here we have our condensed chromatin here on the left-hand side, and over here on the right-hand side was the decondensed chromatin. So this is representing a gene that was actively being transcribed in their cell of interest, right? So this gene is being actively transcribed. They treated the cell with this enzyme called DNase-1 that can basically chop apart the DNA. And what they saw was that the condensed chromatin basically stayed intact because the enzyme was not able to get in here into those active sites and actually, cut those links. Where in the decondensed chromatin, when treated with DNase-1, it was able to break everything apart. So this simple experiment showed that in order for a particular gene to be transcribed, somehow this chromatin has to get into a decondensed state so that the enzymes are able to interact with the DNA. So how does this happen? So gene expression is actually controlled at multiple levels, okay, in eukaryotic cells. And so the rest of next week, we are going to take a look at transcription, translation, and post-translational control of gene expression. So these are all going to have their own set of criteria that allow for genes to be expressed. What we're going to look at today is looking at some additional levels of control that actually have to come in play before the first one here comes in play before any of these happen, and that is this chromatin remodeling. And so this is our focus for today. RNA processing, we've already talked about. And so that was where we were removing introns and adding 5' caps and 3' tails. And we'll touch on this again a little bit when we talk about these transcriptional controls. And there's also another level of regulation, which has to do with how long that messenger RNA transcript, that mature version, is going to be able to stay alive or active once it's out in the cytoplasm. So sometimes that's referred to as the lifespan or the stability of that messenger RNA transcript. And so we'll also take a look at this next week. All right, so this slide here is just an overview, again, of the process of going from DNA to forming our active protein. The difference between this image and the one I showed you guys on Wednesday is that we're starting to put in now some of these control mechanisms, right? So how do we control which genes are going to be expressed and which ones are not? And so, as you can see, the first thing that has to happen in this control process is chromatin remodeling, right? And this is what's going to open up a particular gene sequence from within this chromatin structure that we have. Once we have our gene exposed, we're able to go ahead and do transcription, which we've already talked about, and you'll get your primary transcript. And then again, another part of this gene controlling process is this RNA processing. And so we will talk again about, removing these introns and that alternate splicing that we briefly mentioned back in chapter 17. So that's going to be another way that we're going to be able to control which genes are expressed. And then again, once we're out here in the cytoplasm, we're going to look at the lifespan of how long this transcript can actually last out here before it gets degraded. And this is going to vary depending on the particular gene, right? So some genes we want their transcript to remain active because maybe we need lots of copies, where others we don't want it to stick around for a long time. So there is a lifespan for how long that messenger RNA strand can stay alive or viable out here in the cytoplasm. And then after translation, we're going to see that there's even additional, even after, it's gone through this whole big process, there still may be things that we need to do before that protein can become active. And so again, another layer of gene control here. All right, so we've already talked about, just to review, so this DNA is wrapped around these histone proteins and creates a structure called chromatin. We know that that structure is tightly packed, so much so that RNA polymerase cannot access the DNA because it's too tightly coiled. And therefore, the very first step of gene regulation is chromatin remodeling, where we must be able to decondense that portion of the DNA so that the gene of interest can be transcribed. All right. So some of this information I pulled from the NCBI website to kind of give us, again, just some context here of how many genes we're looking at. All right. So this data was extracted at the conclusion of the Human Genome Project. And so it's called Build 33, and if you click on this link, it'll take you to this website. So what they were able to find in the human genome is that there are about 32,000 genes. And out of that 32,000 genes, about 18,000 of them have specific messenger RNA alignments, meaning that that gene can be transcribed into a messenger RNA transcript. All right, what those other additional genes are, is still under investigation, or they may be, used for something else. But as of when this was posted, 18,000 of them have been confirmed, to have very specific messenger RNA sequence alignments. And when they've looked at these 18,000 different genes that have these messenger RNA alignments, what they have found is that a human gene has about 12 exons that comprise it, at an average length of about 236 base pairs. So when we're looking at, let me go back here. When you're looking at this transcript here of our pre-messenger RNA, what they're saying is that we've got basically 12 of these dark gold regions here are exons, and that the length of this exon on average is about 236 base pairs long. They're separated by introns, and take a look at this. The average length of an intron is 5,478 base pairs long. So way longer than the exons. So that's kind of an interesting discovery, I thought, that, we've got this huge amount of genetic information that, we're going to cut out. It's not going to be part of this messenger RNA strand. So as a consequence, the average intron length is about twice the average length of the transcript, all right? And so the next slide is a table from, again, that website, kind of showing us, here are the number of genes that they were able to discover, so 17,708 with those messenger RNA alignments, meaning, again, I can make a messenger RNA transcript. The average number of exons per gene showed up to be about 12. and the length about 226 base pairs. And then here's the length of our intron and the length of that. So the total average length of a gene was 55,147 base pairs long. All right, so that's a pretty long strip of DNA, right, to make a particular gene. But again, putting it in the context of 6 billion base pairs, I mean, this is really just a nick, right, in the overall genome to transcribe a specific gene. So I thought that information was pretty interesting and kind of puts what we're doing in some context about just really how complicated this process is that's going on inside of our cells. All right, so the next big question, so this is our third big question of the day, how and why are these genes turned on and off? And so if all cells, and we touched on this a little bit, I mentioned this at the beginning of today's lecture, if all cells in an organism contain the same DNA, why do cells look so different? And then how do they perform different functions? And so what I grabbed here was images of different tissues from the human body. So here we've got human skeletal muscle cells. This one here in the middle is skin epithelial cells. And then over here on the right, we have some adipose or fat cells. And so if we look at these, and the reason I picked these is mainly to show differences in nuclei. So each one of these strips here in the skeletal muscle represents a single cell. So a skeletal muscle cell can be about 12 inches long, which is enormous for a cell. And these purple dots that you see kind of within the middle here represent the nuclei. So a skeletal muscle cell can actually have hundreds of nuclei inside of it, which is why they can't actually go through mitosis, because that would be very complicated. But here are their nuclei. In our skin cells, the nucleus is kind of here in the center. It kind of takes up the most of the cell itself, right? We have a little bit of cytoplasm here on the outside, but this big purple blob here represents the nucleus, and you can kind of see that the chromatin structures in there actually. And then over here in the adipose cells, these little red dots that you see are actually the nuclei in these cells. And all of this white that you see is fat. So these cells are responsible for storing our fat, and that fat pushes all of the organelles, including the nucleus, to one side. All right, so 3 different cells, all inside our human body, right? They all look different, they all do different things, but yet each of these cells contains the same DNA. So how does this happen? It's a good question, right? So the answer is controlling gene expression. And so again, these pictures I'm showing you here are data that have been taken from a gene analysis, right? So using a gene microchip. And so each one of these dots over here is representing a specific gene. And green means that it's turned on, and red means that it's turned off. And if we put this under, higher magnification, the brighter, the color, the more there is, the more intense that gene is, being expressed or not expressed inside of that particular cell. And then this is another format, again, where that data is being expressed. And over here, it's listing, the different genes and different things. And I am not, by any stretch of one's imagination, a bioinformatics person. I've not been fortunate to have a class in that myself. It kind of came about after my schooling. And this was not my area of research in grad school, but this is a very, very common and very important field of biological research. All right, so I thought I'd show you guys kind of what one of these little gene chips looks like. So if you wanted to know, if you were studying a particular cell and you wanted to know if exposure to a certain chemical you were doing an experiment turned a gene on or off, you basically, get one of these gene chips and you put it's a very small sample um, on here, and it gets sent off to, in this case, Affymetrix company, and they'll run the assay. And in this particular one, it's kind of using a different type of fluorescence to measure the activity of the genes. But you can see down here the intensity level. If it's showing up dark blue, that the RNA is probably not being, that gene's not being transcribed. Where white is the highest level of intensity, that gene was being expressed at a very high level. And so it's kind of like a heat map or a weather map, right? Where, different colors represent, the intensity of a storm in a sense. And so here where we kind of see like these reds and greens and whites would be showing that that particular gene is pretty active in this cell. So very, very useful and highly sought after research right now is, looking and analyzing this type of data. All right, so now that I've shown you guys an example of like how scientists are able to kind of look at this stuff, let's look at the way that our cells are able to kind of, identify those genes within the chromatin and open it up so that we can actually transcribe them. So we're going to look at 3 different methods. The first method is known as DNA methylation. And so methylation basically means that we're adding this CH3 group onto a cytosine that's next to a guanine. All right, so you'll have this CG sequence somewhere in your DNA strand, and a methyl group will be attached. So the more methyl groups that we have, we're going to see it causes the chromatin to be in that condensed state. All right, so methylation causes, condensing of the chromatin. And so in actively transcribed genes, so genes that we're trying, to get that RNA polymerase to attach to, we're going to see that those particular genes then would have low levels of these methylated, what are known as CPG strands, okay, near their promoters. So this is an important thing to kind of remember here. So actively transcribed genes would have low levels of these methylated CPGs, so this structure here, located near their promoters. Because if they did have that methylation, it's going to keep that chromatin condensed, right? And so if we want to open it up, we don't want those methyl groups present. So if you were looking at your, DNA genome there, you could identify, where these promoters are in those actively transcribed genes by looking for these non-methylated areas. All right, so method 2 is going to be histone modification. And this is going to involve a wide variety of enzymes that are able to add different types of chemical groups onto the histones themselves. And what we're going to see here is that the addition of the enzyme, or the addition of the chemical group that the enzyme adds, can either promote condensation or it can promote decondensation. And so most often in the literature, what you'll see people describe this as it can either turn a gene on. So decondensation would be turning a gene on because we're opening up that chromatin. And condensation would be turning that gene off. All right. And so a histone code of particular modification will then set how tight that chromatin is going to be condensed for a particular gene. So let's look at what some of these modifications are. So the main one is this acetylation of histones. And when we put this acetyl group onto the histone, it usually causes an activation of the gene. So um, this is kind of a funny acronym. So HATs, all right, so histone acetyltransferases. So again, these are a group of enzymes that are able to add acetyl groups onto histones are going to decondense or turn on the gene, right? So they're decondensing the chromatin, which would lead to gene activation. Conversely, if we have histone deacetylases, so HDACs, what this does is it actually removes the acetyl groups. And when you remove the acetyl group, it causes condensation of the chromatin. And so we would see that those genes then are turned off. So lots of stuff going on here, right? I'm sure you guys, your heads are probably spinning. I apologize. So in this picture here, and I know in my textbook, I noted here with where the hat is, I said, well, this is turning on the gene, right? Because I have this decondensed chromatin. So my RNA polymerase is able to come in here and, eventually read this DNA strand to make our messenger RNA. So this would be what, gene activation would look like. And the HDAC, I wrote above this one that this is turning our gene off, right? Because the HDAC is causing our chromatin to basically decondense or to condense. And so that's going to turn off a gene. So this type of chromatin modification, can happen at different times. So this would be triggered by additional cell signaling that would notify this particular gene that it needed to be transcribed. And so an acetyl group could be added here to decondense the chromosome, making it accessible for the RNA polymerase. All right. Any questions? Or be honest, are you guys kind of just like swimming right now with information in your heads? This is, I know, a little bit dense. I will get this uploaded to our site with some questions on there. So if you guys want to go back and practice with this material, I'll have that for you guys sometime in the next couple days. Our third method, and this will be our last method that we look at, is called chromatin remodeling complexes. And honestly, this particular method is not very well understood. They know that there are these things that they've called molecular machines, which basically means we have multiple proteins and enzymes that are doing something to reshape the chromatin. And so some of these molecular machines cause the nucleosome to kind of shift along the length of the DNA. Some of them actually completely remove or knock out the histones from the DNA. And again, their function is to open up stretches of chromatin to allow a gene to be expressed or to be transcribed. All right, and that's about the extent of what our book mentioned on this particular topic, and I wasn't able to really find much more. So for chromatin remodeling, just knowing that there's large complexes of these proteins that are also able to open up the chromatin is all you guys really need to know. Oh no, Carlos is drowning. All right. Why don't we take a break? I only have a few more two more slides for this particular presentation, but it's a little bit more, I think, requires a lot of synaptic transmission to understand. So let's take a break. I can talk about this stuff on Monday because I think you guys will find this part pretty cool if we can kind of just let the other stuff soak in. It has to do with epigenetics. So I know people are excited about that kind of stuff. All right, so is there anything in particular that you want me to kind of go back over while we have a few minutes here? Is there anything I can do to at least get you to get your head above water, or at least your mouth and your nose so you're not drowning? Something I could make clear. Or what are we feeling there? Just, I don't know. I would recommend, if you haven't done it already, that you guys, just take a few minutes to read, section 19.2. It's not very long in your textbook, and I think it will help this make a lot more sense. Okay, so I would encourage you to do that if you haven't already. And then again, if you have any questions, we can certainly revisit this on Monday. All right, one last thing I want to screen share with you guys while we're still together here is I just wanted to talk real briefly here about your Unit 3 project. So our Unit 3 project is found in our Unit 3 tile. And if you scroll down to the bottom here, you'll see unit project. Don't worry about this one. You don't see that one. So this Unit 3 project is about the P53 gene and cancer. And so if you click on this link, all right, it's going to take you to, a page you guys have seen similar before. All right. And this is set up like using the quiz function here in As You Learn. So all of the questions that you need to answer are located here within the quiz. So if I click on attempt 1, oops, that's not what I wanted to do. It's got me in there as an instructor. All right, if I go into preview the quiz now, you're gonna see here are the questions, all right? So it tells you on slide 1, P53 is referred to as these things, and I'm asking you, how are these terms related to each other and to the central dogma of biology? And then I've kind of given instructions what I'm looking for, so you should be writing a paragraph. It should include specific details, definitions, connections between a molecule, a protein, and a gene. All right, and you should discuss what a mutation is. This question's worth 5 points. All right, so you're probably going to want to include more than 2 sentences. All right, so try to explain this the best you can. All right, and so question 2, this one, you'll see that there's, a matching question. And then maybe we have another short answer. This one's only worth 2 points. So here, I'm not looking for a huge, paragraph. A couple sentences would probably answer this question. So pay attention to the point values here for how much effort you put in here, okay? And you're probably going, okay, where are these slides that the questions are referring to, right? So let me go back. So all of the information that you need to answer those questions is found at this link. So the P53 gene and cancer link, this is going to take you to HHMI Biointeractive. And so this is the first kind of slide. All right, that's what I'm referring to as a slide. You can see there's only 8. All right, so this isn't, a super long thing. So you're just going to read the information. Sometimes there's, different things you can click on. There's definitions. There's a video here, so you'd want to click on that so that you can learn about these different things to be able to answer those questions. So again, it's not meant to be super intense, but to give you guys a little bit more background on this information, okay? And then just simply writing your answers right here and As You Learn. All right, so that is our unit project for this final unit that we're doing. It's due on May 1st by 5 o'clock. So that's what we need to do. All right, I have a question here in the chat. What section should we be reading? Okay, so today we covered 19.1 and 19.2. On Monday, we will be covering 19.3 and 19.4, or maybe it's just 3. Let me double check. Yeah, 19.3 and 19.4. And then 19.5 will be next Wednesday. Okay, so I do have the, if you log into Mastering, the reading assignments are chunked in that way, so you guys can, read it there. They've also got some videos built into the e-text that I think would be helpful to watch as well, so feel free to check that out. Does that help, Julie's? Do you have any other questions with that? Great. Anybody else have any questions before we leave for the weekend here? All right. It's probably going to take me till tomorrow to get this lecture posted, just because I'm in meetings pretty much for the rest of this afternoon. So, but I will get that up there. I know this was kind of more complex information, so I'll get that posted so you guys can certainly review that again. All right. I hope you guys have a great weekend. Again, if you have any questions regarding lecture or lab assignments, if you need assistance with an extension, please feel free to reach out, and I can work with you to an extent, okay? We are getting close to the end of the semester, so we do want to try to get everything turned in so we can get it graded. But I hope you guys have a great weekend, and I'll see you soon.